Guys, welcome to my YouTube channel. It's Dr. Kimani again. I studied in Kanga National School, currently pursuing my medicine and surgery course in University of Nairobi. So let's get started. Today we are going to cover the third topic of biology from 1, that is the cell. So we are going to cover the introduction to the cell, the light microscope, magnification, care for microscope, and also magnification power and resolution power. So let's get started. What is the cell? What is a cell? Now remember that almost every living organism, living organisms are divided into two. We have the unicellular organisms and multicellular organisms. Now for the case of multicellular organisms like human beings, there are millions and billions of cells in the body. So now we want to define a cell. What is a cell? The cells make up the human body. So a cell is the basic structural and functional unit of an organism. Is the basic structural and functional unit of an organism. The cell is the basic structural and functional unit of an organism. Many processes that take place in the body usually take place in the cell. Yeah, that's it. So that's basically the definition of the cell, and that's what we are going to talk about in this entire topic, the cell. Now we go to the light microscope. Remember, you have two types of microscopes. You have the light microscope and the electron microscope. But basically, mostly in your schools, you'll get that you're using the light microscope. So basically in this topic we're going to dwell more on the light microscope part. We're going to know what is a light microscope, how does it operate, what are these parts, its functions, and its basic function, the basic function of the microscope. So the basic function of the microscope is to make small organisms or small structures to appear enlarged, for the basis of studies and all that. Now we are going to start on the light microscope. So as you can see here, guys, here is the diagram for a light microscope. So I'll move over to the other side so we can study them more. So as you can see, this is the microscope. So the microscope has several parts. Let's start from above going downwards. So the microscope has two lenses, two types of lenses. One of them is contained here, the other one is found in these structures here. So this is called the eyepiece. This being the eyepiece, inside it will get an eyepiece lens. Just inside here you get an eyepiece lens. And now these are the objective lenses. So the eyepiece lens and the objective lenses contribute to the total magnification of the specimen. So this is the eyepiece. From the eyepiece you have the body tube here. So what's the function? You see this body tube here? Just its function, as you can see, it shows the revolving nosepiece and the objective lens. Yeah, we have the cost adjustment knob just here. Cost adjustment knob. We have the fine adjustment knob also here. The microscope has an arm as well. We have the base. Yeah, we have a mirror. We have a diaphragm. We have a condenser. We have a stage. We have a slide, a clip. Yeah, and that's it. So we are going to look at each function of all these parts of the microscope. So let's get started from upward going downwards. I'm gonna write them here for the cases of your revision. So let's start with the eyepiece. Eyepiece, the eyepiece lens. What is the function of just the eyepiece? That one there. What's the function of an eyepiece? So the eyepiece contains an eyepiece lens that contributes to magnification, of the specimen under view. So guys, that's the eyepiece. It contains an eyepiece lens that contributes to the magnification of the specimen under view. Now we move on to fine adjustment knob. Fine adjustment knob. So what's the function of a fine adjustment knob? As you can see here is the fine adjustment knob. Just here we also have a coarse adjustment knob. So let's start with the fine adjustment knob. So the fine adjustment knob, its work is very simple. It moves that body tube here, this body tube, up or down, up or down. So moves the body tube up or down for short distances. Mark these words here. For short distances. Hence, bringing the image under view into a fine focus. So that's the function of a fine adjustment knob. We've covered the eyepiece, the fine adjustment knob. It moves the body tube up or down for short distances, hence bringing the image under view into a fine focus. Now we've covered the fine adjustment knob. What about the coarse adjustment knob? It's just the opposite. Coarse adjustment knob. What is the work of a coarse adjustment knob? Its work is now to move. So moves the body tube up or down, but for long distances, for longer distances, here you can use shorter for comparison, shorter distances, longer distances, hence bringing the image of specimen under view into a rough focus. Now check here guys, this one is into a rough focus, but this one brings into a fine focus. So using the fine adjustment knob, you'll be able to see the finer details. But coarse adjustment knob, you just have to see an overview. But if you want to see the finer details, you'll use the fine adjustment knob. No, the fine adjustment knob to focus here. So now from the fine adjustment knob, let's have a look at the body tube. What's the function of a body tube? What's the function of a body tube? As you can see there, that's the body tube there. So what's its function? It holds the revolving nose piece and the objective lens. Just that. Or just holds the revolving nose piece, because obviously the objective lens is also held by the revolving nose piece. So it holds the revolving nose piece. And now remember this one here, this revolving nose piece. It rotates, like you can revolve it like this. So when you revolve, you change from one objective lens to another. This one will come here, this one will come here. Many microscopes have 3 objective lenses, or 4. Some have 4. So it will move to 2. This one come here, the other one come there. Somehow, just the way you want. Each objective lens has a different magnification. So depending on the one you want to bring into, to use in focusing the specimen, you can use the revolving nose piece knob. That brings us to the revolving nosepiece now. Revolving nosepiece. So what's the function of a revolving nosepiece? You've just said it. It enables, it holds the objective lens and enables change from one objective lens to another. Holds the objective lens and enable change from one objective lens to another. So that's the function of a revolving nosepiece. So let's continue with the rest. Yeah. Let's continue with the rest here. So we are here at the revolving nosepiece. Now we go to the arm. So we go to the arm. What's the function of the arm? This is the arm of the microscope. It holds the body tube and the stage. This is the stage here, the body tube. It holds the body tube and the stage. Now we go to the objective lens now. So what's the function of an objective lens? So we covered the eyepiece lens. Now we are going to the objective lens. So the objective lens, simply, it works in combination with the eyepiece lens to bring the image of the specimen on a view into desired focus. Works together with eyepiece lens hence contributing to the magnification of the image of the specimen. So that's now the work of the objective lens. Now we go on to the base. What's the work of a base? The microscope has a base. So you can, as you can see, the base is flat and broad. Flat and wide or broad. So what's its function? So its function is very simple. It holds the microscope firmly onto the, maybe you are placing it on a bench or something. So it provides a firm stability to the microscope. Or holds the, provides firm stability to the microscope. As you place it on the bench now, it contributes or it enhances the stability of the microscope. Now we go on to, here is the mirror. Remember I said this is a light microscope. So what's the function of a mirror? So assuming this is light, light hitting here and hitting here. So it reflects these light rays to here. You see? They hit here, they bounce here. So the function of the mirror is to reflect light onto the stage for the illumination of the specimen. So that's the function of the mirror. It reflects light onto the stage for illumination of the specimen. Now from the mirror, we go on to the diaphragm here. You see this is a condenser and this is a diaphragm. So diaphragm has also a very important role here. Its work is to, you see, there may be a lot of bright light that is hitting here. So the function of the diaphragm is just to regulate the amount of light that is going to pass through the condenser towards the stage. So it regulates the amount of light passing through the condenser. That's the diaphragm. Regulates amount of light passing through the condenser. So basically that's the function of a diaphragm. And now we move on to the condenser itself. So what's the work of a condenser? Now you see here, light is coming in many rays from the mirror. The diaphragm regulates the amount of light passing. Now the condenser will be to concentrate, you see? Concentrate light onto the stage. Condenser. Its function is to concentrates light onto the stage. So that's the function of a condenser. Now we move on to the stage here. So what's the work of a stage? So the stage is a flat, it's usually flat like this. Flat. It's a flat platform onto which the slide is placed. You know, the slide contains the specimen that is being studied. So the stage here is a platform. You take the slide, you put in on top there, so that you can use the objective and the eyepiece lens to magnify it. So the function of the stage is it's a flat platform that holds the slide. Flat platform which holds the slide. Now we got to the clip. So you have a clip here and a clip here. So when you put the slide on the microscope, on the stage, you usually have to press it so that it doesn't move, so that you can be able to view it directly. So use the clips to hold the slide. So the function of the clips is to hold the slide firmly onto the stage. Clips holds the slide firmly onto the stage. So guys, you are done with the functions of the parts of the microscope. So I'm going to start back so maybe you can take a screenshot of all of them. Then we move on to the other objective today. The other objective for today. So now guys, remember that the microscope is a very delicate equipment, and also it's very expensive. So that's why we are going to study the careful microscope, and it's usually set also in the exam, because one microscope, maybe the cheapest one currently in Kenya, maybe goes for around maybe between 45,000 and 50,000 Kenyan shillings. So you can imagine having to provide all those for students. It's a bit costly, so that's why you have to study how to care for a microscope. So the first thing, obviously we are going to work with the microscope on the benches. So the first thing when you care for a microscope is that you have to ensure that the microscope is never at any point placed at the edge of the working bench. Never place the microscope at the edge of the working bench. Never place the microscope at the end of the working bench. Why? Because it may fall off, and after falling off, that's gonna cause some huge amount of losses. So never place the microscope at the edge of the working bench. Now from that's the first instruction on how to use a microscope. Moving on to the next. Remember that at times those lenses, at times they may be covered in dust. So what are we supposed to do in case they are covered with dust or something like that? Always use a special lens cleaning tissue for cleaning the lenses, or to wipe out the dust. There's always this special lens cleaning, like there's a specific one that is designed to dust off the dust from the lenses. Always use that one. Now from that instruction we got that one. Maybe okay, it may not apply to all places, but always make sure that you cover the microscope after use. Always cover the microscope after use to prevent it being covered in dust. Always cover. So always cover the microscope after use. Anytime you are done with using the microscope, always cover it to prevent dust from falling off into the lenses. Now the last one you can see, it's about when you carry the microscope in your laps. Always make sure that you carry the microscope with both hands. Now one hand should hold the arm and the other hand should hold the base like this, so that if you carry it in one hand, there's a high possibility that it may fall off. So to prevent that, always carry the microscope with both hands. So now we are done with the care for microscope. You can take a screenshot. I want us to progress to the next objective for today, and that's magnification of light microscope. How do you calculate the magnification of a light microscope? Magnification of a light microscope. So how do you calculate the magnification of a light microscope? Remember I said that a light microscope has two types of lenses, the eyepiece lens and the objective lens. So at any moment you'll be using an eyepiece lens and one of the objective lenses. So if you want to get the total magnification of a light microscope, it's going to be the product of eyepiece lens magnification times objective lens magnification. So this is the formula: total magnification So the total magnification of a light microscope is always the eyepiece lens magnification multiplied by the objective lens magnification. Now let's take an example. Assuming now that our eyepiece lens magnification is times 10, that is magnification, and the objective lens magnification is times 40. So what's gonna be the total magnification? So you take this one here, times 10, you multiply by times 40, which is the magnification of the objective lens, times 10 times 40 times 40 gives you times 400. So this is the total magnification. This is the total magnification. And always make sure that when you're writing magnification for a light microscope, because it's a product, you should have this multiplication sign in front of the magnification, times 400. Without this, you are going to lose a mark. So the formula is usually around 3 marks. The formula itself is 1 mark, the calculation is 1 mark, and then the answer with the multiplication sign gives you the third mark. So times 400. I hope you are clear on that. And also, now that's about the magnification of the light microscope. Now I want us to go to another objective for today. Now that we've seen the care for microscope, we've covered the magnification of light microscope, I want us to go to 2 concepts now. You have something called a magnification power of a light microscope, and you have or just microscope, and you have resolution power. Those are 2 different terms. Magnification power and resolution power. It's going to come in many of your exams, so pay keen attention. So, what is the magnification power of a microscope? What is the magnification power of a microscope? So, magnification power is the ability of a microscope to make small objects appear larger. You see? It's the ability of a microscope to make smaller objects appear larger. Now we go to the resolution power. Of a microscope. Remember, as we progress with this topic, we are going to cover the cell, and the cell has organelles. And so, you get at times that these organelles are so close to each other. But now when you talk of resolution power is the ability of a microscope to distinguish between 2 objects which appears to be very close together as 2 distinct entities. So that's the resolution power. Is the ability of a microscope to make 2 objects, to distinguish two objects which appear to be close together as distinct entities. So now that's the resolution power, ability of a microscope to distinguish 2 objects which appear to be close together as distinct entities. So at many times it's going to become like a differentiator between magnification power and resolution power. Remember to put the words like while in between the definition for magnification and resolution. You can say like the magnification power of a microscope is the ability of a microscope to make small objects appear larger, while the resolution power is the ability of a microscope to distinguish 2 objects which appear to be close together as distinct entities. So that's about it in magnification and resolution power. So maybe we may cover some few things as we wind up. I wanted us to check on, in this topic you are also going to have some, at some points you'll be required to draw the cells, organelles, something like that. So I want us to check on some rules for drawing. Some rules for drawing. Rules for drawing in biology. Rules for drawing in biology. So there are some rules that govern drawing in biology. So now the first rule, whenever you want to draw something in biology, maybe if you draw a cell as it appears under a light microscope. First of all, the cell, that outside membrane or something, it must always have a clear outline. Look at this one. No stoppages. Should be clear outline, no stoppages. The drawing should always have a clear outline. Don't, like draw like this, then stop, then continue. This one here you can't chase around, And also, it should be a clear outline that it's not that you draw this, then you see it's not dark enough, then it says to redraw again, make it just a clear outline once. This one is gonna give you a wrong as well. So you should always have a clear outline. The second one is about labeling. When you want to label, the lines should always be horizontal and pointing direct to the objects that you identify. So the lines should be horizontal and point directly to the object. So that's about the line. And also something else about the labeling lines, they should never cross each other. So labeling lines should never cross each other. Should never cross each other. Whenever you're labeling, make sure that the lines are, they are horizontal and they don't cross each other. That's about labeling. So that's some of just the rules, but there are a lot of rules for drawing, but these are the basic ones. Make sure also the labeling must also be correct spelling when labeling, correct spelling when labeling. Yeah, that's about it. So guys, this was just part 1 of the cell. I'm going to be doing the second part of the cell. Make sure you watch the other one. We are going to continue until we are done with this topic. So you guys have a good time, and you may like, share, and subscribe, and also leave your comments on anything that you want us maybe to clarify. Thank you.